Finally, we're going to talk about two topics, output and regulation. Output relates to the amount of blood ejected from the heart per unit time, and it's impacted or influenced by three different factors we're going to discuss. And regulation is the topic of looking at how regions of the brain help to coordinate heart activity in conjunction with the SA node that actually generates the pacemaker potential. Let's start by looking at output. Output or cardiac output is measured in milliliters per minute. And this is really just a way, of calculating exactly how much blood per minute the heart ejects. And this is a function of two different things. It's a function of stroke volume, and stroke volume is how much blood is ejected either from the left side of the heart in 1 minute time or the amount of blood ejected from the right ventricle in 1 minute time. And we multiply that by the number of beats per minute, and that gives us cardiac output. I'll talk more about stroke volume on the next slide, and we'll see what kind of factors, what three factors can impact or influence stroke volume. Before I do that, though, I want to just briefly mention the term auscultation and what that means. Auscultation is the process of listening to bodily sounds using usually a stethoscope, although you can just use the ear as well. When a doctor places a stethoscope on a patient's chest and auscultates or listens to heart sounds, the two sounds that he's listening to are the lub-dub sounds. And what those lub-dub sounds are indicative of is proper functioning of valves. The lub sound is associated with the closure of the AV valves. As those AV valves close, blood will slam up against the now closed valve, and that produces a little bit of turbulence, and that turbulence is perceived as the lub sound. The dub sound, the second sound in the sequence, is reflective of the closure of the semilunar valves. Again, because when those valves close, blood that's moving slams up against those now closed valves, produces a little turbulence, and that turbulence is perceived as the sound, the second sound. All right, moving on to stroke volume. So stroke volume, as I said before, is a measurement of the amount of blood ejected from either the right ventricle in 1 minute or from the left ventricle in 1 minute. And we cannot actually directly measure stroke volume, but what we can do is indirectly calculate it based on knowing two other things. Remember in my previous lecture, I talked about the end diastolic volume, which is the total amount of blood in the ventricle at the end of ventricular diastole and right before the ventricles contract. Remember too that end systolic volume is the amount of blood left over in the ventricle when the ventricle is done contracting. Well, if I take these two and subtract one from the other, it makes sense that that tells me how much blood was ejected from the heart. Here I'm taking the total amount of blood, the maximum amount of blood in the heart right before it contracts, and I'm subtracting away the amount of blood that's left in the heart after it's done contracting. Obviously, the difference between those two is exactly how much blood was ejected, or stroke volume. We can impact or influence stroke volume in three different ways. The first of which is we can alter what's called preload. And preload is more or less how much stretch we place on the ventricles before they contract. And the way to stretch the ventricles is to fill them with more blood. The more blood present in the ventricles, the more the elastic, the collagen and elastic skeleton of the heart will stretch. And the more it stretches, the more preload we have. It's kind of like rubber bands. If you think about it, the more I stretch a rubber band, the more it will snap when I let go, the greater the recoil. Same is true with the heart. The more I stretch the ventricular chamber with preload, with more blood, the more it's going to contract when it finally does. This is called the Frank-Starling law. And the Frank-Starling law basically says that the more you fill the ventricles, the more the stronger the strength of the contraction when they do contract. The importance of this is that by having the Frank-Starling law in place, I can maintain the right side of the heart and the left side of the heart being able to keep up with each other. If, for example, I have suddenly a vast amount of blood returning to the right side of the heart, like let's say you just started running and you have a bunch of blood returning to the heart, well, now the right side of the heart is going to be handling, relatively speaking, a little bit greater volume of blood than the left side. It can accommodate that because it stretches more, and the more it stretches, the more it contracts. That blood then will get pushed out to the lungs, and as it returns back to the left side of the heart, now the left side of the heart has a greater than normal amount of volume of blood to eject, but that's not a problem because, again, the more volume I have, the more the ventricular chamber stretches, and the more it stretches, the more it's able to recoil. So this helps to keep the left and right side of the hearts equal in terms of their ability to push whatever blood they receive onto the next part of the circuit. When I have a failure of either the right or the left side to keep up with the demands of blood volume it's receiving, I can get a condition called congestive heart failure. And what that simply means is, let's say if the right side of the heart undergoes congestive heart failure, that means the right side is not able to pump all the blood it receives, so blood begins to back up in the vena cava. And as a result, we start to get a buildup of fluid pressure in the entire systemic circuit. And that is played out as an accumulation of fluid in tissues. You probably know that term, edema. Edema means accumulation of fluid in the tissues. So people with right side congestive heart failure, they tend to get fluid accumulation in places like their ankles, in their legs, etc. On the other hand, let's say it's the left side of the heart that isn't able to keep up with the demands, and it's not its Frank Starling ability, its ability to contract when stretched has diminished. Well then, because the left side of the heart receives all of its blood from the lungs, it's the lungs that will start to see an accumulation of back pressure. And so now fluid, the pressure increases in the lungs, and fluid from the blood begins to leak out of the capillaries and into the lung tissue. And these people end up with very wet, soggy lungs. They have a very wet cough. It makes it difficult for them to breathe, and it makes gas exchange in the lungs much less efficient. That's left side congestive heart failure. A second factor that can impact stroke volume is contractility. Contractility refers to how great a contraction strength the myocytes can generate. And be sure that you're careful to distinguish this from preload. Preload relates to recoil after stretching. Contractility relates to the myocytes themselves and how strong of a contraction those myocytes can generate. Principally, it's the autonomic nervous system that influences contractility probably most vigorously. The sympathetic portion of the autonomic nervous system, for example, we know releases epinephrine. And epinephrine can have a positive influence on contractility strength. It can make the strength than it might otherwise. By comparison, the parasympathetic nervous system, through signals it sends along the vagus nerve, that's called vagal tone, by the way, can actually negatively impact contractile strength. Now, when I am tweaking strength of contraction, I am messing with what are called inotropic agents. Inotropic agents. So any factor that alters contractility of the myocytes is an inotropic agent. And you can have positive inotropic agents. Those are ones that increase levels of contractility. So for example, the release of epinephrine would be a positive inotropic agent. And you can have negative inotropic agents, and those are those chemicals or factors that diminish the strength of contractions, such as signals along the vagus nerve from the parasympathetic nervous system. So that's one way we can alter contractility. Inotropic agents are different from chronotropic agents. Chrono means time. Chronotropic agents alter the number of beats per minute, versus inotropic agents, as we said, are responsible for altering the strength of contraction of the myocytes. So I'll give you a couple of examples of inotropic agents, agents that can alter contractility. Let's look at levels of potassium. If a person has hyperkalemia, that means they have greater than the normal number of potassium ions in the blood. Hyperkalemia, it turns out, is a negative inotropic agent. And if you think about it, it should make sense that this is a negative inotropic agent because in a normal situation, cells have a lot of potassium inside relative to a small amount outside. Well, during the repolarization portion of the action potential, the repolarization phase is principally the result of potassium outflow out of the cell. So normally potassium moves down its concentration gradient. That helps the cell repolarize, and thus we are able then to return the cell back to resting membrane potential so that it can respond to the next depolarization that occurs. If I suddenly, though, increase the total amount of potassium out here, I have hyperkalemia, in other words, well, I've altered the gradient. And now when I open those potassium gates during repolarization, I don't get a very vigorous outflow. I get a very meek outflow of potassium because the gradient does not favor vigorous movement of potassium out of the cell. And what that does then is it prevents the cell from repolarizing. And if the cell cannot repolarize, it cannot be ready to fire again. Another example of a inotropic agent would be norepinephrine and epinephrine. And these are obviously released during times of fight or flight. So this is when you want to increase heart contractility. So these would be positive inotropic agents. And another positive inotropic agent would be hypercalcemia. And that is basically when you have higher than normal levels of calcium present in the blood. Remember that the calcium that enters into the heart myocytes is what causes the release of even greater amounts of calcium from the sarcoplasmic reticulum. So the combined effect of that is more calcium rushing into the cell means more calcium released from the sarcoplasmic reticulum, which means greater strength of contraction. The last thing that can impact stroke volume is afterload. Afterload sometimes is a little hard for students to understand, but afterload basically is the amount of pressure of blood on the semilunar valves. So if I were to draw a really simplified version of the heart here, and here are my 4 chambers, you'll remember that coming out of the right ventricle, there's the pulmonic semilunar valve, and that leads to the pulmonary trunk, and then out of the left ventricle we have the aortic valve, or the aortic semilunar valve that leads to the aorta. And after the right and left ventricles contract and they eject blood out their respective blood vessels, when they begin to relax, blood drops by the force of gravity back down over top of these semilunar valves. And that blood puts pressure against those semilunar valves. So that is called afterload. Okay, that pressure of the blood sitting on top of the aortic and pulmonic semilunar valves is afterload. And the greater this afterload is, the harder the ventricles have to work in order to overcome that pressure to eject the next round of blood out. So people, for example, with hypertension will have an increased afterload here. They have greater blood pressure, therefore greater pressure pushing on that aortic semilunar valve, and that means this poor left ventricle here has to contract all the harder in order to eject blood out of that chamber. That's afterload. So let's talk briefly about regulation. And there are a number of different agents which can regulate the behavior of the heart, principally regulating heartbeat, but also, as we saw in the previous slide, you can also regulate contractility. We're going to talk about chronotropic agents on this slide. These are ones that regulate the timing of the heart, the number of heartbeats per minute. I'll give you a couple of examples. One is digitalis, which you may hear of people taking occasionally for their heart. Digitalis is a derivative chemical from the foxglove flower, which is a flower lots of people grow in their gardens. And digitalis is a negative chronotropic agent, meaning it has the ability to slow down the rate of heart beats. So the total number of beats per minute is slowed. It also ends up being a positive inotropic agent. So while it's slowing down the number of beats per minute, it actually helps to increase the contractility per minute. So that's an example of a chronotropic agent. Another one is acetylcholine. Acetylcholine is also a negative chronotropic agent. So, two examples there for you. Again, I want to remind you that the nervous system itself does not initiate the contraction of heart muscle tissue. It's the myogenic cells that do that. All the nervous system does is either up or down regulate the number of heartbeats per minute. And it does that based on information it receives from the cardiovascular control center of the brain, which is located in the medulla oblongata. You'll remember the medulla oblongata is right down here in the brain, and it contains the cardiovascular center. And that cardiovascular center has subparts. Some of the neurons in that cardiovascular center are a part of what's called the cardioacceleratory region. Their job, obviously, as the name implies, is to send out signals along the sympathetic nervous system to increase the overall beats per minute and contractility of the heart muscle. Some of the neurons are located in the cardioinhibitory region, and their job, as the name implies, is to slow the heart rate down. It does that by sending signals along the parasympathetic nervous system's main neuron that goes to the heart, which is the vagus nerve. And we can send signals along the vagus nerve to the SA node, to the AV node, and this causes the release of acetylcholine. And remember, acetylcholine is one of those negative chronotropic agents. Norepinephrine and acetylcholine's modes of action differ, obviously. Acetylcholine causes a slowdown of the heart. It does that by opening potassium channels, by the way, if you're interested. So more potassium moves out of the cell, and so the cell hyperpolarizes. And norepinephrine is a positive chronotropic agent. It tends to speed up heart rate. And it does that by impacting beta-adrenergic receptors, which you learned about briefly in Bio 201 when we were talking about muscle tissue. Those beta-adrenergic receptors that are located on cardiac myocytes are responsive to norepinephrine, and their response to it is to overall increase the strength and contractility of the heart muscle. And if you ever heard of somebody being prescribed beta blockers, what they're being prescribed is drugs that have the ability to block the interaction between norepinephrine and those beta-adrenergic receptors. And so what that does overall is it diminishes the effect norepinephrine has on the heart so that contractility does not increase and so that heart rate does not increase, and we can keep a person's, for example, hypertension in check. We also have some other agents that contribute to regulation of heart behavior. Those include baroreceptors. From 201, you'll remember baroreceptors are receptors that are in charge of monitoring blood pressure. Baro relates to pressure, so like a barometer measures atmospheric pressure, a baroreceptor measures blood pressure within the circulatory system. You have baroreceptors, for example, located in the aorta, and there are also some located in the carotid body. They send signals off to the brain, and those off to those regions of the brain we talked about previously, the cardioregulatory center and the medulla oblongata, and there that information is processed by the medulla oblongata, and if, for example, heart rate gets too high, then signals will be sent along the parasympathetic neurons, along the vagus nerve to slow heart rate down to try to decrease the overall blood pressure. There's also chemoreceptors. Chemoreceptors monitor the types of chemicals that are found in the blood. For example, they monitor levels of carbon dioxide. They can also monitor things like the pH of the blood, sometimes oxygen levels of the blood. And for example, if carbon dioxide levels get too high, then oftentimes a signal will be sent to the heart to speed up the rate of beating, to try to circulate blood through the lungs more rapidly to get more oxygen loaded into the blood. Again, controlled by that cardioregulatory center of the medulla oblongata. Potassium can regulate heart rate. We mentioned previously that hyperkalemia, or too much potassium in the blood, can impact the behavior of the heart. It can cause heart cells to hyperpolarize, for example. And so the heart muscle doesn't contract as readily. It actually has difficulty contracting, and a person can actually go into cardiac arrest because the heart muscle just goes into diastole and it can't contract again. So the opposite of that, hypokalemia, causes a greater amount of a greater amount of stimulus needed to reach threshold, because this also causes hyperpolarization. The reasons are different. Really here with hyperkalemia, it's more about making it difficult for the cell to repolarize. Whereas for hypokalemia, it's more that it's hyperpolarized. I wouldn't ask you that difference on a test. You should know, however, that hyperkalemia leads to cardiac arrest during diastole, whereas hypokalemia makes it more difficult for the heart cells to reach threshold because they're hyperpolarized. There are some hormones that can also impact the heart behavior, the number of beats per minute of the heart. For example, the kidneys, we're going to learn about later, secrete a hormone called renin. Renin is a hormone that indirectly causes the production of something called angiotensin II, and angiotensin II elevates blood pressure. So the kidneys can release renin. Renin increases blood pressure, and that obviously impacts heart behavior as well. The hypothalamus can release antidiuretic hormone that you learned about before. This regulates blood volume, thus blood pressure and heart behavior. And the adrenal glands can release aldosterone, which indirectly conserves water. Remember that it retains sodium, and where sodium goes, water follows. So we hold on to sodium, water volume increases in the blood pressure goes up, and that impacts heart behavior.